Good afternoon, everyone. Welcome to our industry update. I'm Chris Bolus. I'm a hepatologist at the University of California at Davis. Been coming to these meetings for a few years now, so I think a few might recognize me, and I think I recognize some of you as well. It's a real pleasure to be here. I do have to admit I'm missing Ricky and Don this time, but I hope you're all having a really excellent conference. So we've got three industry speakers with us today to share their thoughts and insights in terms of how do we get medications, therapies to patients with PSC. And this is a real challenging topic for us, and as you all know, this is something we've not been very successful with over the years. And I think getting their insights can maybe clarify why this is so difficult for us. Maybe I can start by having some people raise their hands first of who has ever participated in any clinical research. That is, where you had to sign a form, and even if we're collecting a blood sample, stool sample, how many people in the room have done that? Pretty good. All right. How many people have participated in a clinical trial, that is a study where you actually took an investigational drug or medication or procedure? Excellent, excellent, good. So, you know, there are lots of people here that have participated in these trials, lots of people that haven't, and there are lots of reasons why people might want to, and let's do that. Who has not participated in a trial but would like to and hasn't been able to? Yeah, so there's even more people who would like to participate in trials but can't, either because of geography or criteria for these trials. So these are some of the issues we have to deal with. If I can have the first slide then, because I don't have a clicker. Ah, I do. Okay, so why is it important to be in a clinical trial? All right, so I just want to give a real brief overview of how medications, drugs, make it through this whole regulatory process and then get a commercial on TV that you watch. So there's a traditional pathway, and that is where there's some studies that are done in the lab and animal models, and there's some thought that this could be helpful in a disease. It's then tested in a small number of people, oftentimes healthy individuals, just to make sure it's safe and what type of dose should be used. And then it goes into what's called a phase 2 study, again, looking at safety in a particular disease, and again, trying to see what the dose should be. And if it makes it through there, then it has to get through a phase 3 study. And what the FDA is looking for in this phase 3 study is, does this drug or what medication improve how a patient feels, functions, or survives? And that's key. That's what they're looking for. They're not looking to see if your liver tests get better or anything else like that. They want to know, do you feel better, do you function better, or do you live longer? And so we're talking about clinical endpoints here, and so feels could be how your itching improves or your fatigue improves, and we need measures of that, of some way of reproducibly quantifying your symptoms. Or how do you function? Are you hospitalized less? Do you have fewer ERCPs? Or do you not develop cirrhosis? It's complications like ascites, need liver transplantation, have episodes of cholangitis. And these are things that don't happen very often. And so for some diseases that are chronic like this, there are what are called validated surrogate endpoints. And so, for example, for hepatitis C, drugs were approved just because it got rid of the infection. And if you get rid of the infection, then we know people will do better. We don't have anything like that for PSC. Now, just really briefly, I want to say that there is an alternative pathway called the accelerated pathway, in which a drug can get a conditional approval. It can get approval, but then it has to be confirmed in subsequent studies. And this has been used for approval of drugs for primary biliary cholangitis, or PBC. As many of you know, there have been drugs approved in this kind of related disease. But as we heard yesterday, one of those drugs has been withdrawn from the market in the US, and previously been withdrawn in the European Union, because it couldn't be confirmed later on. And as you can imagine, if a drug is available through your insurance company why would you go into a confirmatory trial with a placebo control where you might just get placebo? So it's very hard to run those studies after that. So the FDA has pretty much said this is not a pathway forward for PSC, so we can't go this way. So these are the challenges that we're dealing with in terms of trying to get drugs through the regulatory process so we feel confident that they're effective, that they make people feel better, function better, live longer. But it's really difficult because this is a disease that lasts a very long time, and it is going to be very challenging to find those therapies. So I'm trying to get to my next slide. I thought I had another slide. Maybe not. I do. Can I go back? There we go. So the challenges are, as I said, the endpoints are very rare. They're at low rate, so it's difficult to do this, but we have opportunities. The FDA has expanded kind of what these endpoints could be in terms of development of cirrhosis. And as we'll hear in just a minute, there are treatment options for symptoms, which is a way we can get to drugs to at least make people feel better. So with that as a background, I want to introduce our first speaker, and that's Dr. Joanne Kwan. She's the Chief Medical Officer at Mirum Pharmaceuticals. And Dr. Kwan has nearly 3 decades of experience developing novel therapies, including those for rare diseases. Dr. Kwan got her bachelor's degree in molecular biology at the University of California, Berkeley, and her medical degree at Stanford, followed by residency in internal medicine at Mass General, and a fellowship in pulmonary and critical care medicine at the University of Washington. So I look very forward to hearing from Joanne.
Thanks, Chris. And thanks to PSC Partners for the opportunity to share some of our work with you today. Next slide, please. Or I guess I do that. Okay, great. Our disclaimers. And first of all, who's Mirum? We're a company based in the San Francisco Bay Area. We also have operations in Switzerland, where we run our European and our global operations out of. We're about 350 employees, and we were started in 2018, so we're still a fairly young company. We are focused solely on rare disease, and our main focus so far has been rare liver diseases. A key part of our approach in thinking about drugs and how to design studies and make them available to patients is thinking about patients, working with groups such as you to understand the unmet need, what problem are we trying to solve, how do we design trials, how do we think about endpoints in a way that's meaningful for you, and also in a way that actually takes into account the practicalities of working with FDA and trying to get things approved. And then ultimately, how do we get drugs available to patients, and how do we support them in the process? We have 3 approved therapies in our pipeline and more in development. Let's see. It's not advancing, but other than that. Our first product is called Livmarli, or maralixibat, and it is approved for 2 rare pediatric liver diseases, Alagille syndrome and progressive familial intrahepatic cholestasis, or PFIC. And we're currently working on volixibat, which we're testing in primary sclerosing cholangitis and also primary biliary cholangitis. Let's see, I think I'm going to need some help to advance the slide. It's not quite, there we go, okay. Great. Now, I feel that I shouldn't be telling this to you because you folks know what the symptoms of PSC are. And also, this information comes to us from you. This comes from the Voice of the Patient report and also a lot of work that PSC Partners has done to help us understand what challenges you face with PSC. You know, doctors may think of PSC as a liver disease, but in fact, we appreciate, through talking with you, talking with PSC Partners, how much it affects the rest of your lives. Things like itch and fatigue are 2 of the key things that are important to you, and how do we solve that? As Chris mentioned a moment ago, we view this as an opportunity because the FDA says they will approve drugs based on how a patient feels, functions, and survives. So feels could be itch, could be fatigue, okay? So this is incredibly important information that we use to think about how we can think about the science, think about what problems clinicians face, and then think about the problems that we're trying to solve for you as patients. Now, PSC and PBC are different rare diseases, but they sort of share some commonality in what we call cholestasis. And that's really an impairment in bile flow that causes buildup of bile in the liver in particular, okay? And that is associated with the symptoms such as itch and fatigue and some of the other things that I showed you on the other slide. PSC is more, due to scarring and fibrosis in the bile ducts, whereas PBC is more inflammatory. So, some differences in terms of exactly how they start, but they do share this aspect of cholestasis. And really, that's what we're trying to treat with the medicine that we're developing. Downstream of having cholestasis, you can get liver dysfunction, and then you get a lot of these other symptoms. And so what we're trying to do is to treat cholestasis with a drug that we call an IBAT, or ileal bile acid transporter inhibitor. So in healthy people, the IBAT transporter allows us to make bile and then recirculate it, so you don't have to keep making it. However, in people who have PSC and PBC, you actually have too much bile acid. And so by allowing the body to get rid of it, interrupting the recirculation, we're actually helping the process and helping cholestasis. And so the goal of the medicine that we're developing called volixibat is really to interrupt this, allow the bile to be excreted in the stool, and then hopefully to help the symptoms such as itch and maybe fatigue, and then maybe decrease the damage to the liver as well. So in terms of clinical studies, the study that we just completed enrollment is called VISTAS, volixibat in patients with PSC and itch. This is a large global study. PSC is a rare disease, so we've had to go to many different sites in different countries. It is a double-blind, randomized, placebo-controlled study, and that means that if you sign up for this study, then you're randomized. You don't know, we don't know, your clinician doesn't know exactly which drug you got, whether it's active or placebo. After 28 weeks in the study, everyone is offered what we call open label treatment. So everyone is offered the active drug, and then we continue to follow. We just completed enrollment in this study, and we really want to thank all of you for all of your support. This is not something that we do alone, and this is something that we do with you and for you, okay? Given the timeline of the study, this means that we'll have results in the second quarter of next year, which we're very excited to share with you. And so a big thank you to everyone in the community, PSC partners, all the patients, the entire community, the physician community, the patient community, the caregivers. This, we recognize, has taken a large collaboration, the level of engagement, really gratifying to see the number of people who participated in the study or interested in participating in the study. And we're really looking forward to sharing results with you in this study. In addition, we have another study. This is actually a larger study with volixibat in patients with PBC and itch. So again, similar concept. We're trying to treat cholestasis. Similar design study, double-blind, placebo-controlled, randomized study. This is a bit larger study, so we are still enrolling this study, and we anticipate completing this enrollment in 2026. So again, thank you for your time and your attention. Please join us at our Mirum booth, which is in the hallway. We'd love to talk to you about our studies and our approach, and just love to meet you. Thank you.
Great. Thank you, Joanne. That's really interesting, and congratulations on completing enrollment on your trial. We really look forward to hearing those results and moving that forward. The next speaker is Ben Miller. He's the Vice President and Liver Portfolio Development Lead for Ipsen. And Ben's been specialized in the design and execution of clinical trials and has played a key role in advancing innovative therapies, including elafibranor, in early development, regulatory approval, and commercialization. Elafibranor, through much work by Ben and others, is now approved for PBC conditionally. And they completed a trial in PSC, which I hope he's going to share with us as well. So it's a real pleasure to have Ben here.
Thank you, Chris. And thank you very much for inviting me and for allowing me to provide an update on some of the clinical research that we're doing in PSC. But similar to Joanne, I first wanted to start with who Ipsen is. Ipsen is actually around a 100-year-old French company, but we've only been doing PSC research for a couple of years, so it's very possible you've never heard of Ipsen. I work in our US office, which is in Cambridge, Massachusetts, and we are dedicated to creating better outcomes and transforming the outlook for people living with rare liver disease. And kind of one thing that we often kind of use internally with our meetings is that our focus on liver disease is rare. So our current rare liver disease focus, of course, includes primary sclerosing cholangitis. That's why I'm here today. But we also have, as Chris alluded to, elafibranor, which is approved for the treatment of PBC, as well as other products that are either approved or in the development for some other rare pediatric cholestatic liver diseases like PFIC, Alagille syndrome, and biliary atresia as well. Oh, let me go back. So the purpose of my presentation today is to provide an overview of our ongoing clinical trials for PSC in response to being asked to present this. And I just want to make it clear that the safety and efficacy of elafibranor and rivaroxaban in PSC are just still currently being investigated and are not approved by the FDA. So briefly, what is elafibranor? So elafibranor is an orally administered PPAR alpha delta agonist. And we believe that by activating both PPAR alpha and PPAR delta, that may function synergistically to have a positive treatment effect on the pathophysiology of PSC. And as the schematic on this slide shows, by hitting PPAR alpha and PPAR delta, it decreases pro-inflammatory cytokines, it decreases bile acid synthesis as well as oxidative stress, and also decreases pro-fibrotic pathways. And so all these things combined, based on what we know about PSC, suggest that this product may have potential. And so actually we have an ongoing study with elafibranor in PSC. It's called Elmwood. Actually, the 12-week randomized double-blind placebo-controlled portion of this study is now complete. The results were published earlier this year and are available online. There's also a plain language summary, the details of which are in the bottom of this slide, but it's in the Journal of Hepatology if you would like to learn more. This study does remain ongoing. In the double-blind portion of this study, patients were randomized 1:1:1 to either elafibranor 80 milligrams, 120 milligrams, or placebo. And after 12 weeks, we evaluated the efficacy and safety. And based on the results that we saw, elafibranor 120 milligrams appeared to have perhaps better efficacy and was just as safe as 80. And so for patients who are still in this study, they're receiving elafibranor 120 milligrams once daily for an additional 2 years of treatment. This study was designed previously with input from perhaps some of the folks in this room. We did get patient input and collaborated with PSC partners on this study before we started it. And we ended up including 68 patients from 62 sites around the world. And as I mentioned, this study's ongoing, but enrollment is currently closed. The other product that we have in development for PSC is a molecule called rivaroxaban. Until recently, it was also called A3907. Maybe you've heard of that. It's another orally administered, meaning you take it by mouth. It's a systemically bioavailable apical sodium-dependent bile acid transporter or inhibitor, or ASBT for short. This molecule is earlier in development than elafibranor. So the study that is also ongoing, but enrollment is not currently open, is a small phase 2 study that was evaluating different doses of rivaroxaban in adult patients with PSC. This study was designed so that patients who first enter the study get treated with the lowest dose, which was 10 milligrams once daily. And after that cohort was complete, we look at the efficacy and safety data, and assuming things still look good, then we go to the next dose and so on and so forth. This study enrolled 18 patients. We only had sites in Europe for this study, so those were enrolled in Spain, France, Italy, and Poland. The primary focus of this study is first safety, but we're also collecting a lot of biomarkers to look at disease activity. We did complete this study in July 2025, but we're still going through the process of getting the results. So for this one, I would say stay tuned, and maybe at a future meeting or at a future scientific congress, we'll be able to share results and then determine what future development of this molecule may also look like. And so with that, it's a brief presentation, but I did want to say thank you again for allowing me to present. And the picture's a bit small, but thank you on behalf of the broader team. And this picture is not all, but a decent number of the folks working within our rare liver disease R&D program. And you can see that there's a lot of people working on these drugs across a variety of different functions, working very hard to try to bring these therapies to patients. And so on behalf of them as well, thank you. And with that, I will turn it back over to Chris.
Thank you, Ben. Our third speaker is Heather Powell, and Heather is the head of medical affairs at Esperion. She's been there for a number of years, and prior to that worked at Janssen Pharmaceutical as a senior medical science liaison, and is also served as a trauma step-down clinical pharmacist at Advocate Healthcare and assistant professor of clinical sciences at Roosevelt University. Esperion is relatively new to the PSC world, so look forward to hearing the newcomers on the block.
Thank you. Right. Thank you on behalf of Esperion, particularly the discovery teams and the product development teams. It's such an honor to be invited here to share our new program so early on. So a little bit about Esperion. Esperion was founded in 2002, and we're currently in the cardiometabolic space. So really focused on ACLY inhibition, or ATP citrate lyase. We have two products on the market with the backbone of bempedoic acid, which is a first-generation ACLY inhibitor. This product lowers LDL, and thereby lowering cardiovascular risk. In April of this year, we announced our discovery program and our development program for primary sclerosing cholangitis. So I want to focus on the basics of sort of what our program is founded on in terms of the science. We really are focused on dysregulated cellular energetics driving diseases. So we know that when these energetics are dysregulated, there's a disruption of the TCA cycle that leads to abnormal energy production and utilization. I'm going to bring you guys all back to your high school or college biology classes on the left-hand side with a canonical TCA cycle. Some of you may remember it as the Krebs cycle. So this is where our body's cells use fats and carbohydrates to generate ATP, or energy, for our cells. And this typically occurs in the mitochondria, which is the powerhouse of the cell, if you all remember from high school. Now on the left-hand side of the screen, there's the non-canonical TCA cycle, and this is sort of a sister cycle that was discovered a couple years ago. And this is utilized by highly proliferative cells, cancer cells, stem cells, all of those types of cells that have high energy demands. Now this occurs not only in the mitochondria, but it also occurs in the cytoplasm, which is that space inside the cell, as well as the nucleus. Now I want to draw your attention to, sort of by the bean structure on the right, ACLY, and thereby acetyl-CoA. So ACLY, which is the acronym for ATP citrate lyase, is the key enzyme in this non-canonical TCA cycle. And acetyl-CoA production is the byproduct, or is the product of the non-canonical TCA cycle. We know that when acetyl-CoA production, that it regulates four things, essentially: cholesterol and bile acid synthesis, gene expression, protein regulation, and fatty acid production. So using what we know about this non-canonical TCA cycle, as well as what we know about the disease state of PSC, we started to look at PSC as a disease state that would be viable for targeting ACLY. From this, ACLY upregulation is directly related to multiple mechanisms of PSC progression. And the three I want to focus on are down at the bottom. So cholestasis and injury of the liver and bile ducts, inflammation and immune cell recruitment, and fibrosis in liver and bile ducts. One of the things that Esperion is very proud of for identifying these molecules or our lead candidates in our program is that based on a multitude of data from human genetic studies to multiomics, which looks at a whole host of genes, proteins, metabolites, and human diseases, our molecules were designed and built specifically for PSC. So these are not molecules that are repurposed off of the shelf from another disease state. These ones are specifically built for PSC. And as you can see, there's a rigorous process that they go through during discovery and preclinical development. Now the next 4 slides, I think, really show a picture is worth 1,000 words to me. And these are what really get me excited about these molecules in the disease state of PSC. So the next 4 really follow the same cadence. I'm gonna orient you to it. On the left-hand side, and these are all mouse models, so histology from mouse livers. On the left-hand side, we have a mouse that is essentially otherwise healthy. And you're gonna see a bile duct is sort of that white circle, and then around it, there's collagen, so it's stained red. You can sort of think about that as fibrosis, inflammation. We gave these mice different models to try to mimic what PSC would look like. And so in the middle picture, you'll see that we gave the mice a chemically induced liver injury. So that red looks more angry, right? Moving over into the right-hand side, this is where we've introduced into a mouse our molecule. So ESP1336 plus that same chemical-induced liver injury. And as you can see, there's not as much red angriness on that slide. So ESP1336 reduced markers of liver inflammation as well as fibrosis. Okay. In another model, this is using injury essentially induced through the mouse's food. We looked at a state of progressive biliary obstruction. So again, the control is on the right, or on the left, excuse me. In the middle is where that mouse had injury. And then on the right is where you can see, in conjunction with ESP1336 and the induced biliary obstruction, the reduction of liver enzymes as well as fibrosis. Okay. Likewise, in a model inducing inflammation, very similar results on the right-hand side with reduction in fibrosis as well as markers of liver injury. And then finally, in the most severe model of liver injury, which is bile duct ligation, essentially we clamp off that bile duct and let all of those bile acids pile up. You can see there's necrosis, there's inflammation, there's immune cell recruitment and fibrosis in that middle box. On the right-hand side, when ESP1336 was also given to the mouse, you can see the results when bile duct ligation was given. So based on that preclinical histology data, the next question is, what does this molecule look like? So our lead compounds are currently well characterized, and we are looking forward to daily oral dosing. And right now, we're not anticipating any liabilities into moving into the next stage of clinical trials. So the million-dollar question is always, well, when will this potentially be on the market? Or what does that look like, the first in humans? So I'm happy to say that we are on track, and first-in-human studies will commence in late 2026. Okay. So on behalf of Esperion, we're really excited to start our journey in PSC with you all, and we're really hopeful for potentially having an oral next-generation ACLY inhibitor that targets all 3 of those mechanisms. So thank you, and really look forward to working with you.
So those are 3 really, I think, great talks that kind of span the whole, pipeline pathway that we talked about at the very beginning. We kind of went backwards, I guess, from sort of near the end, kind of in the middle, and then the very beginning. So I think it really illustrates quite well sort of what it takes to get to this and some of the barriers that we face. So I have a few planned questions here, and then I think we'll take some questions from the audience. So Joanne, how can drug developers incorporate the patient perspective, especially around symptoms, into the drug development process? And what approach has Miriam taken to ensure patient-reported outcomes and real-world symptom burden shapes trial design?
Right. I think that's actually, we've illustrated that well in terms of how, our study Vistas was designed. So we chose, as a key endpoint, as the primary endpoint, a patient symptom. And part of that is recognizing it's important to patients. It's important also from a practical perspective because it's something that the regulators, the FDA, will recognize is what we call approvable. It's something that they would recognize and say, hey, this is a meaningful outcome that can be measured and that serves as a basis of approval. So that meets a couple of those needs. And part of it is that we need to understand like what are you as patients experiencing and how do we measure it? How do we go about showing that this is something real? And there's a whole process of what we call validation that we have to go through with the FDA to make sure that this is a robust endpoint, that we're measuring what we think we're measuring, it's reproducible, and that a certain change is meaningful. So there's a lot of rigor that's put around it. There's a whole kind of discipline around it. We have familiarity with that process, and I think that's important that we apply that to symptoms that you tell us are important for PSC.
I think this symptoms are always challenging for us. I think it's much easier for us to send blood to the lab, get a result back, and say, oh, your alkaline phosphatase went up or down. Symptoms is very difficult, and so these tools are really important,
right,
as you say, and how much change is meaningful to patients. Ben, what are the barriers to advancing drugs that have shown positive phase 2 results but have not progressed to phase 3?
Yeah, I think you covered it a bit in your intro to the session, but PSC is a really tricky disease, as you all know. There's a reason there's no approved therapies, and in drug development, the ideal scenario for these chronic progressive diseases is a surrogate biomarker, and PBC is an easy example just because it's within the same family of liver diseases. In that disease, when we had a phase 2 study, we looked at alkaline phosphatase, which is a validated surrogate marker that's known to correlate with long-term clinical outcomes. And so after 3 months of treatment, we saw ALP go down quite quickly, and we had high confidence then going to a phase 3 study of 1 year duration that we could recreate those findings. In PSC, as Chris alluded to, FDA and regulators want to see, does this improve how patients feel, function, or survive? And so as Joanne was talking about, itch, feel, that's one path to go. For elafibranor, we want to focus more on the functions and survives component. And with no surrogate endpoint, our phase 2 study was only 12 weeks. So we saw some nice biochemical responses. We saw some promising things with itch and with fibrosis. But in the end, it was only 12 weeks. This disease progresses much more slowly, and there's no lab parameter that we can do that has truly been shown to correlate with eventually transplant-free survival. So in this specific disease, kind of the leap between a phase 1 or phase 2 study to phase 3 is just about as big as you'll find kind of in drug development. And so, we're working on a phase 3 design, but it's going to be much bigger and much longer with these actual clinical outcomes. Where the phase 2 provides some insight, but it's there's a long way to go, let's say.
Great. So Heather, what early preclinical research, some of which you obviously presented, makes you confident to move your drug into PSC? Are there any key findings there? Was it the totality?
Yeah, absolutely. So, I think the data that I presented was sort of just the tip of the iceberg in terms of our preclinical program. And this work started almost a decade ago by the discovery team, really focused, like I said, on human genetics and understanding the role of ACLY, and then modeling disease with multiomics, with various proteins, genes, and metabolites, and truly understanding that role. We've worked and we continue to work with samples from patients with PSC, and we're confident that with our molecules that we can hit the target of ACLY and that we have an effect on those 3 mechanisms. Like I said, I showed just the tip of the iceberg, but really in the totality, we're really looking forward to moving into phase 3.
Great. Well, we can open it up to questions in from the audience. We have some questions. I can't really see because we've got lights in our eyes up here. Over here, there's a question.
Yeah, I'm wondering for all of you if you anticipate any changes in the FDA in the next few years that could affect the pace of drug development.
I'll let our industry people speak to that, or maybe they don't want to
It's
say
like a hot
anything.
potato.
I always say it's hard to predict with the FDA whether it's going to be easier or harder. I think that to be honest, I mean, they understand that PSC has unmet needs, and they have to balance these two things about safety and efficacy, and they're a bit of a no-win situation in some sense. So I do empathize with the position they're in, and I would say they've worked quite hard, and you know, we've had a lot of conversations both with PSC partners, other investigators, with industry, been quite transparent with what they want to see and why they don't accept certain things that we might think reasonable. Whether there'll be a change in that, I think, is difficult to predict.
I'll throw some optimism into that. I think some of what the FDA has to do is think along with us in terms of, you know, what does this drug look like? What is it supposed to do? Is it providing benefit to patients, and what is the risk? And I think they get better as they think more about it and as they work with us to develop the drugs that we're trying to push forward for you. So I do think that there will be some optimism because, you know, there are drugs that are being developed. There are clearly challenges, but I think, as with many things, you get better if you try again and again, and you have another opportunity to think about it. You think about different angles. So I think, you know, all of this collective effort is really going to be very positive in that sense. And I think we have to be optimistic. I think drug developers, are inherently the most optimistic people you can find, because we always think that the drug is going to work, right? And we have to do our best by you as patients to give it the best chance. So I think there will be a lot of challenges, I think, but I think there is room for optimism here.
Yeah, I agree. I share the same optimism. I'm also an optimistic. I think we see some things from FDA that gives us reason to be optimistic. There's a focus on innovative pathways in rare disease drug development. We hear them saying that they're open to more efficient trial designs, that, you know, maybe we don't need these really long placebo-controlled trials. There will be other ways. And, you know, at least it's a totally different disease, but very recent news from the metabolic liver disease space in MASH that maybe they are finally getting close to a surrogate endpoint there, which would transform drug development there. And seeing the amount of research and collaboration within the PSC community, and now also with the IBD community, to better understand the pathophysiology, it's all trending in the right direction. But of course, with the FDA, like, we don't have a crystal ball, so hard to say for sure.
I'd also plug the WIN-PSC project that I think has already probably been discussed here. And that is really going to be a big benefit for the PSC community to have that data to use both in trials as well as design trials, and go to the FDA and, you know, really be able to design these studies or have surrogates or other markers. So if you have the opportunity to participate in a WIN-PSC study, I really highly encourage that, because it's really going to be important for us.
Thank you for coming. I was recently diagnosed with PSC, and I've noticed my symptoms fluctuate, and my MELD score's been going up and down. And how does that affect how you set up a drug trial when the people are taking this through no maybe effect of the drug? Mm-hmm. Things get wonky.
Yeah.
You want to start? Sure. Okay.
I think that's a great question, and that's something that we need to understand in order to design design a trial that's going to be able to show, is it just variation in the disease, or is it an effect of the drug that we're taking? So, you know, getting as much information as we can from sources such as WIN-PSC or other studies that other investigators have done is really important. And so, sometimes people really focus on, well, can we do a trial? Oftentimes, when we're thinking about doing a trial, we want to understand as best we can what happens with the disease if it's not treated. And that's incredibly important because only on the background of that can we appropriately design a trial. If there's a lot of variation, then we need to account for that variation, and oftentimes it means we have to define very carefully who we're targeting or whether we include more patients. So these types of disease characterization or WIN-PSC is incredibly important, and I really do encourage all of you to participate in that. That is a tremendous help to drug development to understand the disease that we're trying to treat even better. Yeah.
And I'll add that, once a drug is approved, the work doesn't stop on that drug. And I think that's really promising for patients that are not necessarily meeting the enrollment criteria from a randomized controlled trial. There's implementation science projects or, phase 4 projects that we can look at very specific patient populations that maybe didn't meet the enrollment criteria, but are still a large swath of the patient population. And then also, how do we get the drug actually in the hands of those patients? What are the barriers? So, even if you're not a candidate for a clinical trial, the work doesn't stop at approval. It continues for numerous years after.
I think there's a good point regarding the intermittent nature, the waxing and waning of symptoms, particularly in PSC. I mean, we see some of that particularly with itch and PBC, but not to the same degree. And we've recently completed a study where we asked patients, 200 patients with PSC about their itch, and similar to other studies, about 20% of patients with PSC report having itch that's of moderate to severe intensity at the time you ask them. We then ask patients, well, what about in the last 6 months, even if their itch wasn't bad at that time? And then you get up to much larger number, and then if you follow them over time, you see people that had no itch all of a sudden have very severe itch and vice versa. They move all over, and the other thing is that itch is not just itch. The people that itch also have a lot of other symptoms. And so it does make it challenging, though, for, I think, the clinical trials, is because we ask at one time when you come in for your screening visit, what's your itch? And then the instrument we have to use is worst itch in the last 7 days, and it may not have been bad. And so it's difficult to design the trials and know what we're actually dealing with, and then we give it every day, when in clinical practice, again, when we actually get the drug, we may use it quite differently. So I think there's some challenges there, but we have to be kind of regimented in our trials because we need to be as scientifically rigorous as possible. Yeah. Question right here.
We've heard over and over again that pharmaceutical companies don't want to make rare disease drugs, or they don't make money off of it. So, and don't need to be fresh. Why are you all doing this, and how does it ultimately make money off of it?
Yeah, I mean, I think so. What's tricky about rare disease drug development, and I've been doing it for most of my professional career, is when it's really rare diseases, kind of when you hear those stories about not being able to make money in rare diseases, is a lot of times the epidemiology, like just how common or rare a disease is believed to be one thing, and then later on, you start your drug development, 10 years later, then you determine it's actually not as common as we thought. And so that's kind of, for me at least, classically, when you hear those things about, like, you do all this development and then you can't make any money, it's because they got the indication wrong. But rare disease drug development, there is in US and in Europe, like orphan drug designation, there are certain pathways that give you additional exclusivity against generic protection. So it's kind of, every disease is different. Like most things in life, you can't use like a broad brushstroke. Some rare disease drug development does have a good return on investment, and some don't. And it's you just got to do your homework ahead of time before going into what is often a 10 to 15 year development program to bring a drug to patients.
Question back here.
Well, she's given me the microphone. So you guys all sound really optimistic, which is fabulous. So my question is, and maybe number 3, I'm sorry, I don't remember your name, alluded to it. But my question is, are these drugs designed to treat the people who are already like waiting for transplants? They're that sick. If these are successful, can you reverse the disease drastically? Or are we looking at just treating people when their first diagnosis and keep them from progressing?
Yeah, that is an excellent question. So we are at the point in our clinical development program where we're not sure if it will just halt the disease or if we truly have a reversibility component as well. So we're hoping to elucidate that over the next, couple of years. Obviously, we always hope for reversibility for that, but it may just halt the disease if it pans out. But yes, I think that's an excellent question for that. And then, to your point about what type of patient is it, that patient immediately post-transplant? As unfortunately many of you know, typically those very sick patients tend to be excluded from clinical trials. And so in our preclinical work, we are trying to determine exactly at what point in the progression of disease will this molecule potentially have the most benefit.
Yeah, we're often dealing with, if the disease is too advanced, there's safety concerns, number one. Mm-hmm. Number two, it may be just too late. And if we start treating people too early, we won't ever see any improvement because most patients that are in early stage of disease will remain in early stage disease for many, many years. So there's sort of a sweet spot, and when you're talking about a rare disease and you're trying to then just study those that are sort of in this one window, it can be quite challenging. And I think, Ben, was it you that showed you had 62 sites for 68 patients enrolled? That's a small study, Usually it's an average of 1 patient per site they have to open. So these are challenging studies to do.
I've got a question. For really good reasons, post-transplant patients are usually excluded from drug trials. Don't want to rock the boat, don't want to mess up the immunosuppression, all very good reasons. But 20 to 30% of post-transplant patients will get a recurrence of PSC. And then they're going to be almost like a new patient moving forward, but they're also a transplant patient and they're still on immunosuppression drugs. What is the model for eventually testing a drug after you've gone through all the other phases, so that if that patient has to take that or wants to take that drug, they're not taking an untested drug? Does that make sense?
That's a great question.
Yeah, I think it's we wouldn't, just thinking about that unmet need, we know it exists, and so the idea would be not to try to combine the post-transplant population with the pre-transplant because they're just a bit too different and combined into one study. But the idea would be if, at least the way we're thinking about it currently with elafibranor is if we see benefit in the patients with their native liver with PSC, then of course there might be applicability in these post-transplant patients as well, but we would view that as a different study and a different development program.
And similarly for pediatric patients, I think that's the other thing is, there are certain groups that are excluded from certain trials, and that's typically for safety and other reasons.
Thank you all for being here. I've just got a question in relation to all of your drugs, and I know they all work differently, and we're all here for the same goal. Is there any type of collaboration that goes on in the background between the pharmaceutical companies, since you guys also have the same goal as we do?
It all depends.
They do, they just don't want to admit it. So there is something called the PSC Forum, which is a platform that brings together industry, ourselves investigators, PSC partners as part of that, and other patient groups, and as well as the regulatory agencies. So we do get together and discuss a lot of these issues that we're all dealing with. And in particular, I would say for drug development, you know, one of the major barriers we have right now is how do you define an episode of cholangitis? Because the FDA has told us that cholangitis is an endpoint we can use in clinical trials for approval, and it happens relatively frequently. But how do you define cholangitis? If you have a little pain in the right upper side that lasts for, 30 minutes and goes away, and you tell your doctor I had an episode, does that count? Or do you have to go to the emergency room and go in the hospital, get IV antibiotics, get an ERCP, and have them see pus coming out, or somewhere in between? And so that's something that Dr. Trauner, myself, and others have been struggling with, along with industry, to try to figure out and give the FDA what we think is a reasonable definition. And so that's something that's taken some discussion and collaboration with all the groups to get together on. But yeah, there's, you ask a good question, it's difficult.
I mean, I do think this venue and PSC Partners is a great way for us to collaborate, right? We all have different businesses, right? Different focus in terms of what the business is. But one of the things that we can agree on is that this is really for patients. And so PSC Partners is actually a great way for us all essentially to work together with you, to think about endpoints, to think about trial designs, to understand needs of patients. And that's one of the reasons why a group like this is so important. It's actually a way for us to collaborate without us having to figure it all out ourselves. Right? Yeah.
Another example, it's happening again in the MASH space, but could very well see it happening someday in the PSC space as well, as companies are coming together to share all the data from their clinical trials for patients who are on placebo. So the vision being potentially, if we have all companies putting their data together, you now have this really large, rich data set of patients who were treated in these trials with placebo, that then maybe when the next trials come, we all benefit from that data. And it's not there yet for PSC, but that type of collaboration is it happens more in the background. It doesn't make headlines, but we as companies do try to come together in that way as well.
Audra, I can't see yet.
Quick follow-up to that. As a follow-up to that, what would be the barrier to doing that for PSC? That seems like a great idea. The sharing of the placebo data.
I mean, Chris could maybe. I mean,
Well, I think it's something that's been discussed within the forum, and I don't know if in NASH is it through the forum that's happening, or
it is through the forum, and there's a lot more data and longer trials. So it's also there have been studies in PSC as well, but a lot of them, like the one we just did for elafibranor, are relatively small and relatively short, and so with that, it may not be as rich. But yeah, these are things. The short answer is there's not really a barrier, but needs some more thought, and what would that look like?
Yeah, I think that's one of the issues we've had is there really haven't been, trials beyond 24 weeks.
Short.
Most of them are short. There are a few that are longer period, up to 2 years, but they're been only by one company, so there's not really a need to bring them all together. But that's been, I would say, the main challenge, but it's been discussed.
Like everybody said, thank you for being here. I work in IT, and I have a question. AI is all over the place nowadays, and I'm wondering if and how you might be leveraging that. I understand it's great for number crunching and data analysis, so I'm just wondering if you're finding that helpful, or if you anticipate it making a big difference going forward with future trials and whatnot.
I'll say from a drug discovery standpoint, we did utilize AI to screen hundreds of thousands of molecules to look for specific attributes that we thought would lend nicely to our goal. So we use AI in that capacity, as well as other capacities in the company. Obviously, there's guardrails that have to be put around AI, because everything does need to be checked by a human as well. But yes, it's certainly in the drug discovery space right now.
So outside of drug discovery, is there any use for AI in simulated trial design or anything else that you guys are aware of?
I think we're just getting our arms around how we can incorporate it into, our daily workflow, but I do think it's going to increase our efficiency, our productivity. And so maybe we can shorten timelines, maybe we can do more with less. Maybe we can do, a larger trial because we don't have to have that many people. So,
Right, protocols.
yeah, that kind of thing, right? So we're certainly exploring as a company how we can leverage it, and just using it as a way to make ourselves more efficient and get more done in a shorter period of time.
Yeah, it's the same for me in my experience. Like we're looking at ways we can make our own internal processes and steps more efficient, faster. And we've started exploring, but I don't have any examples yet of, we think there's a nice, that AI would also help with finding patients who want to participate in trials, and finding, working in these rare diseases, we think there's potential there as well. And so for our next clinical studies, we want to try to utilize that, but we don't have any success or failure stories yet because we haven't done it.
If we could just stay on the tech side of things, because at the very beginning, asking people that hadn't participated in trials but wanted to, some of the reasons is with a rare disease, matching patients with a physical site is always a challenge. We have patients that fly in from across the country to participate. They're willing to, spend a day or two of travel just to come be in a trial. That's a big sacrifice. And I know there have been models for sort of these virtual visits and schedule things. We've not found them to be very practical for us at our site, but I don't know if you can speak to that or future state of that.
I mean, we've used that in the VISTA study, and I think a number of patients have really benefited from that. And the way we use that is we do have physical sites, but for some patients who may live far from a physical site, what we do is we send the site to them. So we worked with a company who would have a small staff, send someone to the home or to the workplace to do all the evaluations that we would normally, have you and your staff in the clinic do. We think that is actually a great way for us to make trials more accessible to people. We know that to take a half a day or a full day to get to a clinic, park, kind of figure out all the details is an imposition. So we are continually looking at ways that we can make this work. Recognizing that people have families, they have lives, they have children, a lot of things going on. And the more accessible we can make trials and the more we can use some of these newer approaches, we try to do. We have to figure out how to do it. So we want to make sure that all the evaluations are as rigorously done as they are in the clinic. So there's some tricky aspects to it, but we definitely think that that's something we're developing some expertise in, and we really want to explore that more as we do more trials.
I'll add, I actually think that the COVID pandemic lended nicely to a lot of learnings with this. So Aspirion, for example, with bempedoic acid, our currently marketed product for LDL lowering, our cardiovascular outcomes trial was essentially conducted during COVID. So that required a lot of virtual visits and a lot of pivoting by our wonderful clinical development team. So a lot of learnings, I think, can be applied from that period where patients didn't go into their sites, but that we didn't lose patients to follow up, and we continued those trials despite all of the challenges that were occurring in the healthcare system.
Great. So I think we're running out of time here. I'm getting the signal from the boss over there. So I want to thank our three people, panelists from industry who came up and spoke to us, Joanne, Ben, and Heather. I think they deserve a big round of applause.